So let's resume. So just to remind you, DNA encodes RNA, which encodes polypeptides, which fold into proteins. Proteins do chemistry and also provide structure to cells. And cells can either be unicellular organisms of their own or assemble into larger structures that we all would recognize. But this seems like a simple and direct mapping. It actually isn't quite like that. So all of these components, as I alluded to even in the earlier part of this talk, interact with each other in real time in biological systems, including external chemistry and environmental chemistry. So DNA interacts with proteins, RNA interacts with DNA and small molecules in proteins. All of these interact with each other. And this is really more of how the biological code actually works. So shortcuts I'll sometimes use to convey ideas are just shapes. So proteins, this is a protein, a protein with a Pac-Man type arrangement is a protein that catalyzes chemistry, so that's an enzyme. Multiple proteins together is a piece of multi-protein machinery, which is common in life. And I'll simplify DNA or RNA as just a line, and chemicals as polyhedrons. So, one advanced insight is that DNA does not necessarily have a helical structure, and the structure of DNA itself affects how the commands from that DNA are propagated to subsequent biology. So remember I told you that RNA polymerase, a supramolecular protein complex, unfolds a piece of DNA and builds a messenger RNA that encodes protein using that DNA as a template. And remember, this DNA is base paired, and depending on which bases, A's and T's or C's and G's, there's a different number of hydrogen bonds and therefore a different stickiness of those parts of the DNA to each other. So some of you may know in language there's words that are palindromes. And so these are words that are the same when spelled backwards and forwards. Palindromes also exist in DNA, and they often have an interesting biological function in that instead of forming a helix, a complete helical structure, the base pairs can bleb out and make these hairpin loop formations, is what these are called. And so you can imagine if an RNA polymerase is moving down a piece of DNA and runs into something like this, this is going to have an effect on whether or not the RNA polymerase can make RNA by unwinding that piece of DNA. So DNA interacting with itself can regulate biology just by its own structure. And palindromic DNA is one example of that. Another interesting point is that a single section of DNA can encode multiple proteins. And this is because these amino acids, or these base pairs, form three-letter words, and each three-letter word encodes an amino acid, and the amino acids are these letters. But if you shift the frame of coding, for instance, a messenger RNA in a given piece of DNA, you can get three different proteins in a given piece of DNA just based on the frame shift in one direction. But remember, DNA is bidirectional, so you can have three proteins theoretically encoded by one strand going this direction, and another three proteins encoded by a strand going the other direction. Now, this doesn't happen all the time, but this code can be more information rich than just one protein per stretch of DNA. And a good example is a very important enzyme is ATP synthase. It's an enzyme that makes ATP, which is essentially a molecule that drives a lot of the energetic processes in cells. And different parts of this protein machinery, ATP protein 8 versus ATP protein 6, are actually both encoded by the same stretch of DNA, but they're just offset from each other. One other interesting thing that has to do with the chemical structure of DNA that I've alluded to before, but is in fact really important, is that in a given stretch of DNA, a given strand of DNA, how many T's and A's there are versus G's and C's affects both the structure and the properties of the DNA, and how easy it is to make message from this DNA, and how this DNA folds upon itself. So I have right here 3 pieces of DNA, and I've colored A's and T's in black and G's and C's in red. And I apologize for anyone who's colorblind. I just realized that was probably not a good choice in colors. But based upon the GC content of a given piece of DNA, they're all the same length. But if you were to apply heat to melt this DNA, and remember, in real life it has a complementary strand adjacent that's forming a helix. As you increase the GC content, perhaps not surprisingly, one has to melt the DNA at a much higher temperature. And so this difference in melting temperature corresponds to how easy or hard it is to unwind DNA when making RNA from it. And so this also affects how DNA is processed into message and then protein. And a great example of this, this is a hot spring at Yellowstone National Park. These colorful organisms that you see around the edges of this hot springs, or the edges of this pool, contain what are called hyperthermophiles. And these organisms have evolved to live in this extremely hot environment, and maybe not surprisingly, their DNA is often enriched in G's and C's and depleted in A's and T's, because at these temperatures it wouldn't form helices or have much structure if it was too AT rich. And the converse is true in other types of organisms. So environment and evolution can affect this message, and the message's enrichment in one type of base pair versus another affects how a polymerase can turn the DNA into RNA and thus make protein from it. So hyperthermophiles often have to copy DNA with high fidelity, and the reason for that is that their DNA is in an environment that's prone to melting. And so polymerase chain reaction, modern polymerase chain reaction, uses enzymes to unwind and copy the DNA that come from extremophiles, including extremophiles that live in hot, deep ocean vents. And so this is an interesting case of DNA and the machinery used to copy it evolving in a way that's useful to humans in biomedical research because it's very high fidelity due to the conditions in which it has to work. So this is one interesting example. DNA enzymes copying enzymes are error prone, and the proneness to error in the molecular machinery that copies one piece of DNA and makes a daughter section of DNA from it varies from organism to organism, and this is also in fact affected by environmental conditions, which I'll talk about later. But when you copy a piece of DNA, you can have mutations in the base pairs that encode the DNA, but if you recall, there's a great degeneracy in the words that encode amino acids in a polypeptide and the A's, T's, G's, and C's in those words. So this particular stretch of DNA encodes this particular peptide, isoleucine, proline, and so on. This mutant piece of DNA has one base changed, but because of degeneracy in the code, it still encodes the same protein. But another mutation switches the identity of the terminal amino acid in this peptide to a histidine. So there can be both silent and silent mutations where the code changes, but the code's product doesn't change. And then there are mutations that actually change the amino acid code. But what's interesting is if you compare these two pieces of DNA, the RNA made from them might have a slightly different structure, even though they encode the same peptide. The speed at which, or the ease at which the DNA is converted to RNA can be subtly changed even by silent mutations. And then mutations themselves can be beneficial, neutral, or deleterious, and to an organism or overall system. And then these propagate through generations, and this drives evolution. But there's an element of randomness in the copying of parental DNA to child DNA, as it were, and this sprinkles in the random chances for evolution in the process. So one important thing that's happening in the biotech world is making use of extremophile DNA and the proteins that are encoded by that extremophile DNA for industrial purposes. So laboratory supply companies like New England Biolabs and Biorad have been using extremophile polymerases to copy DNA for purposes like PCR and various molecular biology work in the laboratory for a long time now. Big chemical companies like DuPont and many others use extremophile enzymes to help them make chemicals, catalyze the making of chemicals in high temperatures or pressures or in other extreme conditions where you want a version of an enzyme that holds up under industrial synthesis conditions. There's a number of pretty advanced startups that have made their name in this space as well. So companies like Zymergen and Ginkgo and Amyris make, among other things, flavors and fragrance molecules, probably some pharmaceuticals now. Certainly Amyris got started in the pharmaceutical space. And they also use extremophile enzymes sometimes to help make their products because these enzymes hold up better under extreme conditions. And then there's some really interesting early stage startups such as Cinder Bio, which specifically builds biosynthetic pathways from enzymes that come from extremophiles. So the DNA is very stable. The proteins that are made from this DNA tend to stay folded in high temperature conditions. And companies like Invizyme, which is another synthetic biology startup, specifically makes lipids using some of these extremophile gene products. But DNA doesn't only encode messenger RNA. So DNA also encodes promoters and essentially landing pads for molecular machinery that converts the DNA into messenger RNA to land and start processing down the DNA strand. And then also there have to be sections of DNA that instruct the polymerase to stop and disengage because one doesn't want to read through to another gene. So there's punctuation in the genetic code, as it were, and there's specific sections of DNA that encode that punctuation. And then in translation from the message to the protein, a given messenger RNA can contain multiple protein coding regions that get turned into separate proteins. So it doesn't all map, one piece of DNA to one message to one protein. There's actually quite a bit of editing that can get done in between. One example of regulation of DNA itself is that DNA can be modified by attaching chemicals to it that aren't normally part of it. And there's various enzymes that do this, and these are proteins that catalyze chemistry where small molecules get attached to DNA. And if you can imagine a polymerase, either one that makes copies of DNA or that makes RNA from DNA, moving down a DNA helix and then encountering a chemical that's attached to that DNA, that can gum up the works, and it can slow or stall this polymerase and cause it to disengage. And then whatever RNA is being made or whatever copy of the DNA is being made truncates early or isn't completed. And so this is one of the ways that biology is regulated. And obviously DNA encodes these DNA modifying enzymes. So there's all this interaction in a given biological system. In higher organisms, DNA isn't stored in chromosomes. So this is a superstructure of DNA. So if you imagine a DNA helix, it's complex because in a given stretch of DNA, two non-adjacent pieces might together encode one protein and then have non-coding DNA between them. And so this is another level of complexity. And a helix of DNA can be wound around proteins that form these balls called nucleosomes. And then these themselves are wound into chromosomes. And so there's this huge complexity in how DNA is wrapped up and stored. And there's enzymes that modify the proteins in the nucleosomes themselves or the DNA wrapped around the nucleosomes that affects how easy it is to unwind DNA for it to be copied or turned into messenger RNA. And so you can think of this like scrolls, where you can unroll scrolls to a certain section to do reading, but the whole scroll isn't accessible at one moment necessarily. And so there's a vast amount of chemistry that happens that regulates what DNA is accessible for additional chemistry to happen. And so some of these enzymes, like this magenta one, are enzymes that switch genes on, and so these can allow the spooled up DNA to open, and then chemistry to happen, whether it's copying or other modification. So SWE/SNF happens to be a gene complex that's been well studied in this context. Other sorts of modifying enzymes lock the DNA so that it can't be unspooled, and this silence genes because the genes can't be transcribed while they're wrapped around the histones. And so there's a whole regulation just at this higher level of folding of DNA. Now, this actually has been increasingly put to therapeutic use. I'd highlight that very recently a company called Epizyme has a first-in-class drug that is a small molecule that inhibits one of these enzymes, a methylase that acts on these histones around which the DNA is spooled. And so this actually affects the division and propagation of certain classes of cancer cells, first an approval for sarcoma, and then just I think maybe in the last month there's been an expansion to treat a type of lymphoma. So one can actually intervene in biology merely by intervening in the manner in which DNA is wrapped around histones and whether it's accessible for transcription or not. And there's many other people working on this and really interesting literature in this space. Another interesting example is of the external environment having an effect on gene transcription and translation is the presence of vitamin D in an organism combined to a receptor protein. That receptor protein can bind to another protein, and together this complex can bind to a piece of DNA and specifically stick to a part of the DNA that prevents it, in some cases prevents transcription, in some cases enhances transcription. And this allows many genes to be expressed, turned on and off, and these affect metabolism, immune function, and others. So, even just the serum concentration of vitamin D in a human has numerous implications regarding how that particular human's DNA is converted into RNA and thereby into proteins, and has effects all throughout metabolism and health. And then to make things even more complicated, many RNAs don't encode proteins. So Earlier in the talk, I mentioned tRNAs, transfer RNAs, and these RNAs are used to help build proteins from amino acids in the context of, or catalyzed by this enormous mixture of protein and RNA called a ribosome. And as we saw earlier in the talk, this ribosome, which I previously showed as just a blob, is actually a very complex macromolecule made up of several individual proteins and several pieces of mRNA that in this case form a structure and are involved in catalysis of building a growing polypeptide. So RNA itself can do chemistry. Another way that RNA can do chemistry is in the form of ribozymes. And a ribozyme is a piece of RNA that acts like an enzyme in that it itself can catalyze chemistry. And some of these ribozymes catalyze the chopping up of messenger RNA, for instance, to make different pieces of messenger RNA. And if you imagine that this messenger RNA was encoding a given protein, if it's been affected, or if it's been, say, cleaved by a ribozyme, now you're going to get a different, shorter protein made from this message. And then, as I alluded to earlier, a single messenger RNA can be cut up and reassembled to make different proteins. So imagine a piece of DNA that encodes these various exons. These are portions of the DNA that encode RNA that gets turned into a single polypeptide, even though they're non-continuous in the DNA. But what you can have, which plays a huge role in biology, is what's called alternative splicing. And that means that these different exons can be reassembled into different proteins depending on the order in which they're put together and how many of them are assembled. So this given piece of DNA can actually make different forms of related proteins, but not the same protein. So protein A, protein B, and protein C are all made by combining these exons differently. And this alternative splicing happens at the level of RNA editing. So the slicing and dicing of messenger RNA to make modified messenger RNA, which leads to different proteins being made. So one interesting thing is that some organisms, and especially cephalopods, actually have a very stable genome. So their DNA gets copied very efficiently and doesn't change much compared to other organisms. However, compared to other branches of life, cephalopods can edit their RNA much more than most other organisms do. And so there's this interesting trade-off where their genome doesn't evolve very fast, but they can make changes to the proteins that genome encodes on the fly by heavily editing their RNA. And actually there's a hypothesis that the unusual intelligence of cephalopods for animals, for organisms of their, at their position on the tree of life and with their lifespan may be as partially a function of their ability to make really diverse proteins from a single bit of DNA through extensive RNA editing. So that's a hypothesis that's quite interesting, and I encourage you to read about it if you'd like. DNA can travel between organisms, so DNA is not confined to a single organism. These are two bacteria that are transferring a plasmid, which is a circle of DNA, a little piece of DNA that's circular from one bacterium to another. Now, this can have no impact, no significant impact, or it can have a huge impact if this plasmid, for instance, encodes a gene that allows a bacterium to break down penicillin. So DNA being traded by organisms or taken up from the environment by given organisms can really affect that organism's ability to survive selective pressures. And we see this with drug resistance in bacteria, which is an enormous problem now. And let's just say that this plasmid encodes this protein, which you see beautifully rendered in rainbow colors here. And this is a metallo-beta-lactamase enzyme that chews up this molecule which is a penicillin analog and is usually an antibacterial, unless the bacteria have gotten their hands on a plasmid that encodes an enzyme like this. Then they can chew this up and survive treatment with that penicillin analog. So, and then at the level of cells and organisms, there's, for all the reasons that I previously mentioned, there's numerous potential errors that can happen as the DNA code is turned into instructions for chemistry. And this can be direct DNA damage, for instance, from ionizing radiation or UV radiation from the sun, if you like to sunbathe. There can be problems with DNA repair genes, and then alteration and modification, depending upon, for instance, how much vitamin D one gets. So there's a lot of feedback in biology that all comes back to how it's encoded. And cancer in particular is a really difficult system to study because of the heterogeneity. So even a given mass of cancerous cells, they can be closely related, or they can eventually diverge from each other so much that manipulating their biology is not as easy, in that one can affect some cells in a tumor but not all of them. And so this heterogeneity is reflective of the complexity of these systems and is one of the great difficulties in, for instance, treating cancer. So Donald Rumsfeld won't be remembered, well, he'll be remembered for a number of things, but many people don't know that his famous quote is really liked by many scientists, independently of his politics. And so I like to think about biology as sitting in buckets of known knowns, unknown unknowns, and unknown unknowns. And I would hypothesize that right now it's mostly unknown unknowns, so we don't know what we don't know. So I hope I've conveyed to you that DNA code is chemically complex, and it encodes both data and chemistry. And one thing I'd like to bring your attention to is that the world of understanding how this DNA encodes biology is accelerating. Humanity is accelerating in its ability to do these experiments, to ask, what does this piece of DNA encode? And the reason for that is that DNA reading has been cheap for a while, so that's DNA sequencing. But it's now become very inexpensive to write DNA, and it's rapidly becoming less and less expensive to print DNA. So now scientists can look at any piece of DNA that's previously been sequenced and say to themselves, I want a copy of that to be synthesized, and I want to test what proteins it makes, for instance, in my own lab. And you can pull out a credit card and go to companies that will synthesize pieces of DNA for you. Interestingly, this also has considerations in biosecurity. It's much easier to engineer DNA now that one can order it. And so there's really interesting work done around making sure that these technologies aren't used for bad purposes. So I hope I've convinced you that instead of DNA to RNA to protein, there's actually more complex interplay than we've learned in Biology 101. And in these interactions, there's much opportunity to intervene in biology. So some takeaway thoughts: DNA is not destiny. Identical twins who have different lifestyles have divergent fates. So your environment does affect what genes are turned into what proteins, and which DNA is transcribed and translated, and this affects your fate as an organism. Everything in biology is a complex system. Precise engineering of biology requires understanding relevant mechanisms. It understands actually doing the experiment. And I think that's becoming easier and easier due to cheapness of making DNA and laboratory automation, instead of graduate students and postdocs pipetting by hand and wasting most of their time transferring small volumes of liquid into other small volumes of liquid. And the design, test, build cycle, the iteration, is something that we will accelerate our learning about how to engineer biology. And I just leave you with, nature is filled with treasures that evolved in deep time. So if you were to ask me what is the world's most valuable natural resource, it's the DNA that's evolved throughout the world, throughout different organisms. And we are constantly discovering useful molecules in organisms as diverse as marine sponges and plants, many of which were used in traditional medicine. We find what medicinal molecules they make, and they've evolved under selective pressures that we can't reproduce in the lab to do chemistry that in many cases humans wouldn't imagine. And so to me, the greatest resource that's available to humanity, and one that we have to preserve, is this DNA code that encodes chemistry that humans would not think of. Companies are looking for these natural products, are finding these natural products in nature, in part because DNA is cheap to print. Companies like Hexagon, BASF, and Loto Therapeutics are looking for therapeutic molecules made by microorganisms in nature and have engineered other organisms to scale up the production of these. And so our next antivirals, our next antibacterials may very well come from nature. And I think with infectious disease now on everybody's radar, as it probably should have been before the COVID epidemic, but now most certainly is, I think that there's going to be an explosion of people who prospect in the DNA of the world for unique chemistry. And science reveals unique complexities that we don't even really begin to understand. So people think of the human gut microbiome, but there's microbiomes everywhere in nature, and plants have a microbiome too. And I just include the schematic to bring that to your attention. There's very complex plant microbiomes, and a number of companies are trying to understand and manipulate what microbiomes grow with what plants, and especially how can one inoculate plants with a microbiome that, for instance, helps them fix more nitrogen from soil and thus require less fertilizer, or allows them to be enriched in a microbiome that fights off fungus that causes crop rot or something else that's deleterious without using chemical small molecule pesticides. And so there's a whole interesting world out there in terms of opportunities to engineer complex biological systems and to sort of reprogram the environment. So with that, I'll take questions, and I thank you for bearing with me, and I hope I've given just a small introduction to the levels of complexity that flow from the genetic code.